This is my.
There we go.
All right, so welcome everybody. It's really my great pleasure to introduce Dr. Emily Gibson. I'm going to kind of give you the standard I guess this would be the website biography. So Emily's associate professor in the Department of Bioengineering at the University of Colorado Anschutz Medical Campus, so just down the road from us. She has a joint appointment in the neuroscience program. She earned her PhD in physics from the University of Colorado at Boulder with a specialization in nonlinear optics, and she was subsequently a National Research Council, National Academy of Sciences postdoctoral fellow in biophysics, where she studied protein dynamics with nonlinear optical spectroscopy. And then since becoming a faculty member, she's focused on development of optical technologies for clinical applications in biomedical research. But I really want to, so that's kind of the textbook, but I really want to brag about Emily, just the diverse contributions that she's made, both in terms of technology and science throughout her career. So it's been always an exception to work with Emily because she's done things like develop new ultrafast lasers, but then gone on to use these tools that she's developed, ranging from microfluidics to the neurosciences, well, you name it, to not just, so she creates these new technologies, but then really applies them to fundamental, answering fundamental science questions, which is just fantastic. So with no further ado, I'm going to turn this over to Emily. So welcome, Emily.
Thanks, Jeff. Thanks for the nice introduction. And I had the opportunity to work with Jeff when I was a postdoc and learned a lot about microscopy, actually. From working in Jeff's lab was my first experience. So let me figure out how to share.
If you hover your mouse at the bottom, there should be a green, oh, there you go, you got it.
Yeah.
Perfect. I'm gonna mute myself again now.
Okay. So everybody can see that, all right? Okay. All right, so yeah, so I was gonna talk today about some of the work we've been doing at the Anschutz Medical Campus, particularly our collaboration with researchers in neuroscience, Dr. Diego Restrepo, who's a professor of cell and developmental biology, and Dr. Ethan Hughes, who is in physiology and biophysics. And so I want to make sure that I thank and acknowledge these collaborators. And also we've been working closely with Victor Bright and Juliet Gopinath, who are in engineering at CU Boulder, and as well as Mark Siemens, who's in the physics department at University of Denver. Okay, so let me figure out how to advance this. Okay. So mainly, you know, coming from a background of physics and optics, I was interested in developing more imaging techniques, particularly using multiphoton microscopy methods. And so coming to the Anschutz campus and meeting a lot of people in neuroscience and starting to learn more about neuroscience, there's some really cool kind of optical challenges to be dealt with there. And so in neuroscience, you know, people are trying to image the brain and neural activity in the brain. They can do that in animal models, of course. It's difficult to do that in people. So using these types of fluorescent sensors, so you can monitor neural activity by change in fluorescence from cells that are expressing these types of indicators for voltage or calcium. And so calcium concentration changes when you have neural activity, and you can actually visualize the cells as they're firing. And so people have done a lot of work, you know, and trying to mimic, you know, activity. For example, on the left, you see this is a mouse running on a floating ball. So the mouse can kind of feel like it's moving around even though its head is fixed under the microscope. But if you really want to study certain types of neurological, problems or understand, for example, social behavior, navigation, or more complex behavior like how to navigate in an odor plume for mating or finding food, these can't be done with the mouse being head fixed. I mean, it's just not possible. So a lot of the current challenges is, can you do similar imaging, but in a miniature format, in an animal that can just move around freely with some lightweight microscope attached to its head. So that's what I was going to be talking about today. So just to give you an idea, so this is one of the first papers demonstrating a miniature wide field microscope. And so on the left, you're seeing the mouse moving around in its cage. And on the right, you can see the image that's created from this wide field microscope. So this is showing fluorescence changes from calcium concentration using a GCamp reporter. And so those are the kind of images that you get out. And then if you start to do some analysis, then you can see how these neural patterns are encoded. In this example, this is showing how the location of the cells corresponds with the position of the mouse in its cage. Okay, so just to cover some of the previous work on these miniature microscopes. So on the left is the microscope that was in that previous work I just showed. So it's just a wide field microscope. So it uses these miniature CMOS cameras, just a light emitting diode and some optics. And then you can record basically using one photon excitation fluorescence imaging. So recent work has developed some two-photon miniature microscopes. So this is just an example of some microscopes where you're actually using pulsed infrared light that's coupled into a fiber, and then you have to scan your laser focus at the sample and collect the fluorescence light back. And so this is an example here where you're scanning with a miniature MEMS mirror. You're scanning the light that comes in, with a miniature MEMS mirror and collecting the light that's coming back through a fiber. Or another example was demonstrated using like a piezo scanner on a fiber. So you can scan the light that way. Okay, so the reason that you kind of want to go to two-photon imaging as opposed to the wide field imaging, there's a number of reasons, but one is that you can get better penetration depth in tissue. The other is that you get this kind of intrinsic cross-sectional imaging. So if you're imaging through tissue with wide field, you're just going to collect fluorescence throughout like the axial region of your image, and that will obscure a lot of things, as is shown on the left here. And on the right, you're just imaging at the very focus of your microscope objective, so you can resolve in much finer detail. And so, okay, yeah, and so again, I'm sure, if you're in the department with Jeff, you've known a lot about how multiphoton imaging works. So, on the top is showing fluorescence from one photon excitation. So you get excitation throughout the whole volume. And with two-photon fluorescence, you only get excitation at the very focus of your objective. And so, yeah, so the other advantage is that you get enhanced penetration depth in tissue. So, so this is just taken from a paper from Chris Chu's lab. So this is looking at the effective attenuation length of light as a function of wavelength. And as you can see here, you know, as you're going to longer and longer wavelengths, you're going to get less scattering just from Rayleigh scattering. Although you do have to include absorption of water and biological tissue. But there's a few places where you can actually, you know, get better penetration depth. So at 1300 nanometers or at 1700 nanometers, you get enhanced penetration depth compared to single photon. Okay, and yeah, so just as an example, so this is effectively if you're doing one photon excitation, so you're using really visible light, green light at 550 nanometers, you know, you can only really get through 70 microns before you get a lot of scattering. But for two photon, you can get down to 130 microns effective attenuation length. Okay. And so another thing that that we've been investigating that some of these other microscopes haven't shown before is the ability to actually take three-dimensional images through the tissue. So the previous work I showed you was just, you know, imaging in one focal plane, just imaging the hippocampus. And in many cases, you know, you're not going to have just one flat structure of the brain, right? It's not going to magically all be, you know, exactly in your field of view of your focus, but you might need to image in a volume to really see all of the activity because there's many complicated structures in the brain. So for example, this is showing an image, a two-photon image of what's called the olfactory bulb. So this is cells that sort out, you know, your sense of smell. So how can you recognize like banana from skunk, let's say. So this kind of recognition and processing is done by these cells in the olfactory bulb. And as you can see, there's many different layers of cells that are involved in all of this computation. And so the ability to image, you know, through the brain and image these different cells is really critical. Okay, so when we were investigating how could we, you know, build such a microscope, we looked at different methods that could actually change the focus. So ways of doing non-mechanical scanning of the focus. And so there's several different sort of optical elements that are used for that in microscopy. So one is this tunable acoustic gradient lens or TAG lens. But, you know, in this one, it can actually change your focus very rapidly. However, it's really not compact and it has takes a lot of electrical power, so it's not suitable for fitting onto a mouse's head. Another type of lens that people often use for microscopy is a pressure-driven tunable lens. It's called OptiTune. And so this one, you know, it does have a compact size, but it does have a lot of optical aberrations, and it's importantly, it's sensitive to the orientation of the lens. So if your mouse is moving around, that's not going to work very well. So finally, there's a type of tunable lens called an electrowetting lens, and that kind of meets all of the requirements that we were looking for to actually miniaturize and put onto a mouse. And importantly, it's impervious to any kind of orientation or motion, uses low electrical power, and can be made very compactly. Okay, so I can figure this out. Okay, so yeah, so an electrowetting lens, actually, it's got some pretty cool physics that goes on here. It's like something that you could kind of do a simple freshman lab demonstration. So it's an effect that was first observed, you know, couple hundred years ago. So if you have a conductor and then some kind of hydrophobic layer on top of the conductor, and you have a drop of water on top of that, so normally the water will kind of, because it's hydrophobic, it'll form like a bead. If you apply a voltage bias between that water droplet and the conductor, then you can actually control the contact angle, right? So the angle at which this water droplet has with the surface. And it's a very robust and sort of reproducible fact. So you can just change the voltage and then change that contact angle. And so because it kind of operates as a capacitor, it doesn't take a lot of current. And so it can be made very low power, and you can use it for a bunch of these different types of kind of optofluidic devices. So to make this electrowetting lens, in effect, you kind of have your drop of water, and on the side you have your conductor coated with hydrophobic layer like Teflon, and then you put on top of that, you put like oil, and then as you change the bias, so there's a conductor on the bottom that's in contact with your water droplet. As you change the bias between that and this sidewall conductor, then you're changing that contact angle and you're creating this tunable lens. And so this is a cool demonstration from Juliet Gopanas and Victor Wright's group, where they did this in a glass tube. So you're changing that voltage, and you can see how it changes the curvature. So effectively, it's a tunable lens. Okay, so what we did is we designed our miniature microscope. It's a fiber coupled microscope, and this is the design that the student Barish Osbey came up with. So we basically have our fiber that delivers the light. It's collimated by an A-sphere, passes through the electrowetting tunable lens, and then it's focused by these two objectives onto the sample. And so this is just like a Zmax ray tracing diagram showing what happens when you're actuating that electrowetting lens. So it can basically image in 3D volume. And we actually get a range of about 200 micron scanning range. Okay, so this is just showing a photo of this microscope. So it's made with a 3D printer, and then we have our fiber coupled to it, and then it's adapted onto a microscope objective, which is nice because then you could basically, if you have a multiphoton microscope, you can just take off the objective and then screw on this adapter, and you've turned your microscope into a fiber coupled microscope, that was our plan. So this is basically what it looks like attached to the mouse. So what you do is you have to do a surgery. So we basically, sorry for the physicists who don't like the biology side of things. I know that when I start to talk about the mouse surgeries, like our collaborators in electrical engineering, the students get pretty grossed out. But anyway, so you do the surgery, you inject the virus in the brain, that expresses these fluorescent calcium indicators. And then you basically replace the skull with a cover slip window. And then we attach this base plate onto the head. And then you can click on or detach the miniature microscope. And so this is just looking at, so through this microscope, you can see with wide field the fluorescence signal through the cranial window, and these kind of dark regions there are from blood vessels. I also want to mention how we do the scanning. So basically, I've told you about how we change the focus position axially or in Z. But to do the lateral scanning, we decided to use these imaging fiber bundles, they're called. So there's only one company that sells these types of fiber bundles. They're used often in endoscopy and biomedical endoscope applications. So from Fujikura. So basically, it's a fiber that consists of many cores, and the cores are coherent. So if you put in light, on one side, it's relayed exactly to the other side of the fiber. And so the one that we've been using has a 0.5 millimeter diameter and about 10,000 individual cores. Each core is about 2.4 micron diameter, so they're pretty small cores. So 2.4 microns, and they're separated by about 4.5 microns. So what we do is we just attach this fiber onto our microscope, and then the microscope itself has a galvo scanner. So we just scan our beam on the surface of this fiber, and it couples the light into the different fiber cores. And so what you actually see from this is you get a very pixelated image, right? Because you're only getting signal from the cores and not from the surrounding. So we have this pixelated image, but then we can, do some processing and retrieve the effective signal back. But basically, the lateral resolution of our microscope is really determined by this fiber bundle. Okay. And so another issue I wanted to talk about that we run into, so these endoscope fiber bundles are not meant for multiphoton imaging. So it's kind of a challenge to try to do multiphoton imaging through a meter-long glass fiber. So one of the things that happens when you do this is you get dispersion. So you're trying to propagate a short laser pulse through a 1.5 meter piece of glass. So if you take the pulse coming right out of your Ti-sapphire laser, it's about 70 to 100 femtoseconds in duration. And so if you look at that has a certain spectrum. Let's say it's about 15 nanometer spectral width. But after you go through this 1.5 meter piece of glass, then each of the different frequencies in your spectrum propagates, with a different amount of time, has a changing index of refraction. And so what you get out is a pulse that's stretched out in time. And so in that case, then, you have a pulse that's stretched out in time, has a lower peak intensity, and then that reduces, your multiphoton excitation efficiency. So how you can get around that is using what's called a grading pair. So this is a way using 2 grading pairs where you send a pulse in and the different spectra are dispersed and reflected off of a mirror and comes back out. So if you do this, just the geometry of it, so if you look at the path length from the geometry in this case, you can actually arrange it so that the bluer wavelengths will travel faster than the redder wavelengths. So in that case, that's called a negative chirp pulse. And so if you put this negative chirp pulse now through your your glass fiber. The fiber applies positive dispersion. So here the blues are coming earlier than the red, but in the fiber, right, the blue light travels slower than the red light. And so at the very end, then they can all line up together. So that's what we initially tried for this. And it worked fine until you get to higher laser powers. So what happened here, if we started to go above about 4 milliwatts of power, suddenly we saw that our pulse was getting stretched out more and more. And it turned out that what was happening is that our spectral the width of our spectrum was actually narrowing. And so that ends up being due to sort of the nonlinear effects. Okay, so since I'm talking to a bunch of physicists, I wanted to throw this in, which I don't for anybody at the Anschutz Medical Campus. But because you are my people, the physics people, so just to share what's going on here. Okay, so what's happening as we're increasing the power is that we're getting sort of this nonlinear effect. So your index of refraction of the material is not a constant, so it's not just n, but now it's n times the intensity of our light. Okay, and so what happens there? So, if you look at the phase of your electric field, you have your center frequency times time minus kz. You substitute what k is, right? 2 pi n over lambda. And now this n depends on the intensity. So what we see here, right, is where we are in this pulse in time. We're seeing this change in intensity. Okay, so we know that, right, the frequency, right, the frequency of our pulse depends on the time derivative of the phase. And so now that depends on, the index as a function of intensity. So what's basically happening here is that this is the front of the pulse. So over the front of the pulse, as the intensity starts to increase, right, the center frequency starts to decrease because it depends on negative dn dt. And then at the tail end of the pulse, it's the opposite. So intensity is decreasing with time, and so your frequency is gonna increase. And so just naively, what's happening here is that if you send in a pulse that's negatively chirped, so the blue arrives earlier than the red. Basically what happens here is that it shifts the frequency, right, at the front end, shifts it to a red side, tail end shifts it to the blue side, and that'll narrow your spectrum. Okay. I think I need one more slide to really convey that message, but just to give you an idea. So that's kind of roughly what's going on and why you see this spectral narrowing when you put a negatively chirped pulse into the fiber. So then how can you overcome that? So effectively, this is kind of a brute force method, right? So what's happening is we're sending our negatively chirped pulse into the fiber, we're getting that spectral narrowing. But instead, we could actually add another fiber before the grading pair and use that to broaden out our spectrum. So if you send in a pulse that's basically transform limited into a long fiber, it's going to broaden the spectrum from cell phase modulation. And then we run it through this grading pair, which will apply you know, a negative chirp, and then run it through the other fiber, and then that'll narrow it back down again. You know, so the idea is to kind of balance it. So you're going to broaden it and then reduce it, and it'll end up about the same as it was initially. Okay, so when we did that, and we looked at you know, different power levels, we did find that we could maintain, you know, a short enough pulse. Okay, so this is sort of the first build of this, what we call the pre-compensator. So we've got our single fiber, and then we've got our grading pair, and then we sent it into this kind of custom built multiphoton microscope right there. So, okay, so this is doing some of our experiments here in the mice. So just so you have an idea of how it works on this mouse in particular. Yeah, so the mouse is wearing the miniature microscope attached to that imaging fiber. And there's another electrode that's controlling that electrowetting lens. And so the mouse eventually, it kind of gets used to it, used to having that fiber attached to its head. And then mice, they kind of like to run a lot. So if you put a wheel in their cage, they'll start running on it. And they can run for like miles and miles. So this guy, eventually he starts running. We'll see. Eventually in this video. There he goes. He starts running. Okay, so that's what these experiments actually look like. Well, this was doing sort of our test, right? So, so this is some of the results we got from the fiber microscope. So this is basically, you know, looking at a single plane and watching, you know, the fluorescence activity from GCamp. So. Okay, so that's kind of what it looks like. This is kind of the post-processed image. And then this is sort of what you see when you're changing the voltage on the electrowetting lens. So you can really scan sort of through, that's 190 microns. So starting from sort of the top layer and down 190 microns deep. Okay. And then this is sort of a processed image of activity that was taken in the mouse. So this is at different focal depths. And you can actually correlate the activity. There's software that does this where you can look at different regions of interest and then plot those in time and correlate that with motion. So in this case, we were kind of in the sensory motor cortex, and we did find some correlation of motion with activity. Okay, and here's another recording we did. This was actually in the piriform cortex, which is involved in, again, odor processing, because I work with Diego Restrepo, who studies olfaction and odor. So this is a deep brain area, so we used a like a very small diameter Grin relay lens that's implanted first into the mouse brain to access this deep brain area, and then placed our fiber coupled microscope on top of that to image. And so this is actually looking at an image of a female mouse with this male bedding. So when the mouse approaches the male bedding, then you see a lot of activity in the piriform cortex. And when it's away, you get less activity. So there's cells really light up there. Okay. All right. So the other advantage, you know, compared to some of the other miniature microscopes are the fact that you can actually do multiple color imaging, because we're basically using, you know, the multiphoton microscope, laser source, and detectors to basically for excitation and collection of the light. So here we could image two colors. So these are two different types of cells. One is labeled with green fluorescent protein, and the other is a redder shifted fluorescent protein. And so these different types of fluorescent proteins kind of can be genetically targeted to different types of cells. So we were able to show that we can do that with our miniature microscope as well. And the other thing that's kind of cool about this type of design is that, you know, you can basically change the electrowetting voltage as you're scanning, and that can let you actually change the angle of your focal plane that you're doing. So this is basically doing a scan, and you're able to kind of rotate that focal plane by plus or minus 30 degrees. And that could be very useful, again, because, you know, you might not have a brain structure that's perfectly flat, but something that you want to image at an angle. Okay. And another thing we've been looking at is how could we do sort of two-plane, multi-plane imaging? Because again, you know, like in the olfactory bulb, you have kind of two different cell types in two different planes. So if we can quickly image both of those simultaneously, that would be of great benefit. So we were able to show that we could adjust, you know, between two planes. These are only separated by 30 microns, but theoretically we could do it, you know, further to image different cell layers at about 5 hertz, which is good enough for the dynamics of calcium. Okay, and finally, so with this, you know, again, like I said, you know, these types of coherent imaging fiber bundles are really not designed for multiphoton imaging. In fact, you know, mainly they're used for kind of white light imaging or single photon fluorescence or autofluorescence. So what happens is that if you just try to do imaging through this fiber, if you're using one photon excitation, you actually get very uniform fluorescence light back. But if you do it with 2 photon, you get this like really inhomogeneous. So some of your fibers are almost completely dark, and others have higher transmission. And then it turns out that the reason for that is that basically, you know, the different fibers have different dopants in them that actually changes, you know, the index of refraction. And so they actually do this intentionally to prevent core-to-core crosstalk. So if you're doing like 1 photon imaging, you don't want, you know, it to like cross into another fiber. But that causes a big problem with 2 photon imaging. So we're working on ways, talking with Jeff as well, of ways that we can start to correct for that problem. And then you can see some other kind of interesting effects going on, again, with this like core-to-core fiber coupling, and how it seems like higher order fiber modes couple outside of the main fiber that you're exciting. So there's definitely a lot of improvements sort of that we believe we can make to this. Okay, and so I also wanted to just mention some other work that we have done. Again, this is the collaboration with Juliet Gopinath and Victor Bright's labs. And so with them, you know, they actually make their own electrowetting lenses, and they do the processing and manufacturing for that. And so it turns out they were working on these electrowetting prisms for beam steering applications. And so what happens with that is that instead of just having the same electrode on the sidewall around that lens, you can actually have two separate electrodes and apply different voltages on that. So effectively what happens is, you know, you can first apply a voltage equal on both sides so that your contact angle is at, you know, 90 degrees, and that's called sort of the critical angle. And then you can basically now adjust the voltage on the right side higher than the voltage on the left side, and you'll form this prism effectively. And so if you're shining a laser through that, then it'll be deflected. Okay, so the idea, what we wanted to try is, can you use this little miniature electrowetting device as a scanner, and supposedly a scanner for a miniature microscope. And so they manufactured these prisms. This was just shown with like 2 electrodes. If you wanted to scan in 2 dimensions, you could do 4 separate electrodes. But this is the design they came up with initially for 2 electrodes. So as you change the angle, or as you change the voltage, then you can change the prism angle. And okay, yeah, and so, you know, again, this was to replace some of these other kind of miniature scanners, but it would have advantages because it's non-mechanical scanning. Okay, and so we set this up on our microscope just as a proof of concept. So we basically added some optics in, you know, to our multiphoton microscope. So we basically placed the electrowetting prism conjugate to our galvo scanner, and then we used the actuation of the prism to give us scanning in one direction, and the galvo, we just used one of the galvo scanners for another direction. Okay. And so this is kind of what it looks like when you're scanning it on the card. So in this case, the galvo is providing us the fast scanning axis, and the electrowetting lens was providing the slower scanning axis. Okay. And this is just some more info on those lenses. So we initially tried it just with a fluorescent calibration slide. So as you see that when you're changing the voltage, it's actually changing the steering sort of in a nonlinear manner. So you get, you know, this distortion in the image that you get out, but you can correct for that kind of with post-processing. And then we applied this to cultured cells. So this is looking at cultured hippocampal neurons labeled with GFP. And so we took an image first with the galvo scanners, and then this is showing the corrected image using that electrowetting prism scanner. Got remarkably good results. So we're working right now on, you know, or they in Juliet and Victor's lab are working on making it a 2-dimensional scanner and finding ways to kind of increase the scan speed. And that really kind of has to do with the choice of liquids that you put in there. Okay, and I guess I'm getting close to the end, but I just wanted to also just mention another project that we're working on. This is a collaboration with Juliet Gopinath and Mark Siemens at DU. So what we've been working on is developing a miniature fiber-coupled STED microscope, because basically Juliet and Mark Siemens have done a lot of work with making these kind of donut modes propagating through fiber that could be used in STED microscopy. So we've been working on that. So basically super resolution microscopy, is another kind of really interesting development in biology, because with these types of techniques, there's several types of super resolution imaging techniques. You can actually see below the diffraction limit of light, which is typically, diffraction limit is about 200 nanometers, but you can see below that to roughly 40 nanometers. So you start to see things, on the order of, individual fluorescent molecules, where you can separate sort of two different fluorescent labels, or you can see, dendrites and dendritic spine morphology, or other small structures in the brain. So super resolution techniques have also really taken off, and they've been, kind of critical for a lot of understanding of basic biological processes. But they haven't been really applied much to in vivo imaging, because it's a challenge, I would say. So we wanted to take up that challenge, because NSF gave us some money, and see how far we could go with it. So our idea was, can we make, can we generate the STED mode in a fiber, put that onto a miniature microscope that we could attach to a mouse and do in vivo imaging. Okay, and so STED microscopy, again, that's one of the super resolution techniques. I'll just kind of briefly go over how it works. So this first example is what happens with a diffraction limited confocal microscope. So you basically can focus your light down to the diffraction limit about 200 nanometers with a high NA objective. And then you excite all the fluorophores that are within that region, and then you get sort of this broad, fluorescence emission out. And with STED, what you do is you have your confocal, laser focus excitation spot, but then you overlay that with this donut mode, okay, where it's got intensity on the outside, but then no intensity in the center, and it's at a different wavelength. So that different wavelength actually causes stimulated emission depletion. So it's basically taking the excited fluorophores, and you're shining light at the fluorescence wavelength, and you're rapidly depleting them back to the ground state. And so you can basically just turn off those fluorophores, and then you only get excitation at the very center of the donut, where the STED light is 0. And so your resolution, instead of being lambda/2NA, now you add this additional factor. So it depends on the intensity of your STED beam on the fluorophore. Okay, so how do you create these kind of donut modes that you use in microscopy? So the way people do it on a standard benchtop microscope is using like a phase plate, so that can affect the spatial phase of the beam, and then it focuses to a donut mode. But you can also do this in a fiber. So if you launch different order modes in a fiber, so these are different modes here. So instead of, like your standard nice Gaussian mode, you can launch a higher order fiber mode. So this left one is one of the fiber modes you can launch. and then you can launch another one like this. And if you combine the two, then you get basically a donut. And so the trick that they came up with, Mark and Julia came up with, is if you take those two fiber modes and you actually delay them in time, they won't interfere with each other. And then you can actually bend the fiber and you'll still maintain that nice donut mode. So if those two are actually interfering, that'll destroy the mode. Okay, so we use this technique in a fiber, and again, this is just a proof of principle demonstration. So this is at the focus, looking at the STED beam out of that fiber, and then the excitation beam. So this is looking at the focus in X and Y, and then how it looks in X and Z. Okay, and so using this fiber, we took images with and without the STED beam, and we saw enhancement of resolution. So just looking at a line out here, we could see enhancement in our resolution. So we got down to about 90 nanometers in this case. So yeah, so the next step, basically, you know, we did this proof of principle, and now we have to develop the sort of miniature optics and then test it out in our mice. Some of the stuff that we're interested in looking at, again, is like, you know, the dendrites and spine morphology and how those change as the animal is undergoing different kind of behaviors. Okay, so, oh yeah, so this is another, the past one was the bead, and this is actually an image of a tubulin labeled sample. Okay, so that was all I had to talk about today. I just wanted to, you know, mention the people that were actually in the lab and did all of this hard work. So basically, there were two of my PhD students, Barish Ozbey and Greg Futia, who really did all of the two-photon fiber-coupled microscope development work. Omkar Subakar, who was a student in Victor Bright's lab, did the work together with Barish on, you know, the electrowetting devices. And Connor McCulloch is a new student. I didn't talk about what he's been working on, but because that would have taken too long. So, okay, so I'd like to thank everyone, and I didn't put it down here, but I should also thank the funding agencies, so NSF and NIH, who supported this work. Yeah, and then finally, so we're just, since I'm talking to a bunch of physics students and undergraduate students, so if you're interested in applying for, you know, getting a PhD, I think like CU Anschutz is a great place to go if you're interested in this kind of working sort of in collaboration with neuroscientists and optics people. So if you're interested, please contact me or Diego Restrepo. And we're only, what, 20-minute drive with no traffic away. So yeah.
Awesome. Well, that was amazing. Thank you so much for that talk. I really enjoyed that. We actually have a huge number of questions. So
okay.
we can do this one of two ways. You can either click the Q&A down in the bottom right, or I can just read them to you. So the students have the ability to upvote and downvote, and so do the faculty, the question. So students, feel free to, I guess, just upvote your favorite question. Okay. So if you want to get on the list.
Yeah, I can't see it somehow.
Okay, so I'll just read it.
Click the Q&A.
Yeah,
yeah, if you want to read it.
Yeah, so that sounds good. Okay, so we have some questions. One's from Daniel Scarborough. What are the scan times for the 3D images generated in the olfactory bulb?
So yeah, so again, you're very limited on, you know, because we are doing like a point scanning method, you know, you're kind of limited basically to, you know, a single plane. You can image it in about, let's say, you know, 15 hertz frame rate, to kind of get the full field and the resolution you need. So you're really limited by, right, you're scanning a point focus across the sample. But that's enough for, you know, the calcium imaging. So these calcium sensors, or the calcium diffusion, is kind of a slow process. So, so that's enough. But yeah, so if you want to get multiple planes, we've kind of shown that. But you get, you know, you get hit by another factor of 2. So we could take the 2 planes in about 5 to 10 hertz frame rates. But there are like other ideas, some ideas that are coming out on how to do these multiphoton imaging faster. But right now that's our limitation with this device.
Oh, nice. Great, great. Okay. So another one from David Schmidt here. So the electrowetting prism surface didn't look quite flat in the images. Are the wavefront aberrations from that limiting, do they present a limiting factor in your system?
Yeah, they do. So yeah, with that, it's basically, you know, you do get those aberrations near the sidewall. It is flat kind of in the center of the aperture. So yes, that is definitely an issue, and you can really only sort of operate it if you underfill the lens.
Awesome. Great. And then from David Goldberger, we have, what are the temporal and spatial resolutions, axial and or transverse, required for these brain studies in mice? And are they substantially different than from that of humans?
Wait, sorry. So, are the temporal and spatial dimensions different?
Yeah. So do the resolution that you need vary
Oh,
differently across different animals, like widely?
so, I mean, the size of the neurons is the same effectively. They might be, I mean, I'm not the neuroscientist, so I should probably not be answering that question. I think they're similar size. You know, also the sort of the, you know, the dynamics, the action potential operates similarly. I do think the firing rates of individual neurons differs a bit
I see.
between mice and humans.
Okay.
Um,
Awesome.
yeah, but there are definitely differences. I mean, one is the human brain is so much larger that makes it a challenge, and especially for people who are doing studies on non-human primates, that makes it a real challenge to do these types of optical imaging, and also to be able to image like the full brain structure, which is also larger.
Yeah.
You know, so in those cases, people have been using these grin lenses to kind of image deeper brain regions. But the mouse's brain is actually remarkably small, so it's only like a centimeter by a centimeter. So, so you can access the majority of it with multiphoton or particularly like 3-photon.
Okay, awesome. And so from John Sersky, is the electrowetting lens spherical, or is it's got some other shape? Is it closer to parabolic?
That is a good question. Yeah, I mean, I don't know the answer off the top of my head. So it would be, yeah, so it's basically, you know, you kind of solve, it's like a minimization problem where you're solving the electrostatic interaction and chemical interaction. So I don't know that off the top of my head. There is a good, so COMSOL multiphysics, you can actually model this.
Cool.
If you'd like to. I guess everyone here probably knows that, but knows, you know, those modeling programs, and you can actually, they actually have, I think, an example on COMSOL multiphysics.
Okay.
Yeah.
Cool. Are you up for a few more?
Sure, why not?
Okay, great. So, yes, sorry. This is probably the most questions we've had all semester. This is fantastic. Okay, so from Scott Hunter, can the bubble in the electrowetting lens be broken by vibrations, say from the mouse? And by broken, I mean something like when you take like a level and you shake it, how the bubble can burst.
Yeah, so it's, well, so yeah, that is the case. Like in a level, you know, that's really, you've got fluids with different, let's see, they have different densities, right? So that's how the level actually works, is you're looking at a fluid with a different density within another fluid. But for the electrowetting lens, you actually match the density of those two liquids. And so that's what the magic that makes it like impervious to like vibrations. So you can like shake it and you can turn it, and it's not gonna move.
That's amazing. That's really cool. Okay, so from David Goldberg, can you use the multicolor fiber that you use for scanning to make STED modes? And if not, why?
Yeah, that's an interesting question. Yeah, I mean the main issue is that it's a matter of like how tightly, you know, you can focus that mode. So with the multiple cores, you could never, you know, it's kind of individual kind of sources, and you can never really focus it down tightly enough to be like a STED mode, which is kind of why we do it sort of in a single fiber.
I see. Okay. So, and another, I think, a related question from faculty here, Chip Durfee. So, so I guess he's wondering how coherent the bundle is. So, could you transport like a space and time focused pulse down it? But I'm guessing, based off of what you just said, the individual fibers are not super coherent with each other.
Oh, yeah. I mean, so there's, I guess that's not talking about phase coherence. So that just means, so when it's called a coherent fiber bundle, that's just because, like, you know, the arrangement on one side is the same as the arrangement on the other side. But physicists, when you hear coherence, it takes on other magical meanings. So, but there's, yeah, so the fiber can be phase coherent if you don't touch it, you know, so so if you have the fiber and it's like stationary, you know, you can measure it and you can actually map out, you know, the different sort of phases of the different cores, and then you can actually use that to create, you know, interference patterns at the end of the fiber. But for our application, we can't have a rigid fiber that's not moving.
Right.
So we can't really do anything that kind of you know, relies on like the coherence effects.
I see. Okay. Okay, and then I think we just have one more if you're up for it. Oh, we just got another one. So what's the maximum frequency that the electrowetting scanner can scan at?
So yeah, it's interesting. So there's a lot of different effects that go on. So it depends on the liquids that you're using. So the densities of the liquids. It also depends on the diameter of the lenses. So turns out the smaller diameter you can make it, then you can get very, very fast response times. Another interesting effect with these lenses is that you can drive them on a resonance. So if you take that kind of standard commercial lens and you drive it at a certain frequency, it's kind of resonant. I think at like 70 hertz, it'll operate on resonance. So it's a really interesting, like, kind of fluid dynamics problem where you're exciting these different like drum modes.
Yeah.
Yeah.
Awesome. And then I guess, do you want to do one last one? We just have one more.
Yeah, that's fine. That's
Okay.
fine.
So this is from Randy Lemons. Is there a benefit to using combinations of two higher order, the Hermite-Gauss modes, to generate the Laguerre-Gauss STED rather than a mode converter mechanically bonded to the, it seems like he's indicating a polarization maintaining single mode fiber, but I'm not sure that's what you actually use. But So is there some advantage to doing the two different modes, the two different HG modes, rather than some kind of mode converter at the end?
Oh, yeah, I think okay, that's a very astute question. Yeah, so there is some work that's been done kind of using these phase, a phase plate. There's actually, so, in Stefan Hell's lab, the guy who invented STED microscopy, so they've made phase plates that it'll shape the beam at the STED wavelength, but not the excitation beam. So it's a special customized phase plate where you can put both beams through and only the STED beam gets shaped.
Oh, that's interesting.
But it's not very miniaturized, and it's really expensive if they'll even sell it to you.
Right.
So yeah, but so kind of, the option that we have, which, again, you don't have that kind of heavy phase plate material, is to do it just all through the fiber itself.
Okay, awesome. Well, I have a selfish question. I was wondering, to what extent do you see 3D printing kind of being a helpful thing for designing and constructing these microscopes going forward, or is that not something that you kind of even need to explore?
No, I think definitely 3D printing has become really important with these types of things. I think it's been really helpful for kind of developing different prototypes. You don't have to go and machine each prototype. As you're iterating on your design, especially like for how we're designing how to attach it to a base plate. And some of these commercial 3D printing companies can do very good tolerancing. So that's really been a benefit. It's also, lightweight material, and it's pretty rapid to get it done.
Oh, that's cool.
Yeah. So no, I think that's been a big thing. I don't know, the students in my lab, they really like 3D printing, and I get kind of upset when like all of our optics mounts are being 3D printed. They think that's a good idea, and I'm like, no, that's not a good idea. That's not very stable. You're going to be, aligning things all the time.
Right. So. That's amazing. Awesome. Well, thank you so much. That is by far the most questions we've had in a row like that in a colloquium. So that just kind of goes to show how interesting what you're doing is. So yeah, thank you so much.
Well, if anyone's interested, please contact me. We're always looking for physics students who want to branch out and learn bioengineering and neuroscience.
Awesome.
So, yeah.
This was awesome, Emily. Thanks so much. It's really great.
Okay.
Totally.
Thank you.
Yeah, good talk.
Bye. Stop share. Okay.